We are about to start our third section in today's epigenetic, chromatin, and RNA. And our first speaker, although from far, Geneviève Almouzni from the Institut Curie. And she's going to tell us about chromatin dynamics and cell fate from a histone variant viewpoint that we, until now, haven't heard anything about histone variants. So please, Geneviève.
Thank you. Thank you very much for the opportunity to be with you in video, although I would love much better to be with you physically and enjoying the chit-chat that we could have and getting to see each other. So my sincere apologies. So since I will be talking about the H3 variant and there hasn't been much about it, I will give a bit of an introduction. But first, I just want to get to my first slide. I hope you can see it. If it's switched, do you? I hope so. So the idea is to tell you about how we can learn from histone H3 variant a number of aspects related to genome function. And there's really an important aspect to have in mind is this balance and equilibrium as sort of illustrated by this street art picture from Paris, which I do like. So what I'd like to do is first to give you a little bit of a background, considering histone H3 variant and their chaperone as the players like bricks and architects. Then I will tell you about aspects related to how you can follow new and old histones. And this is in particularly interesting when considering situation when you disrupt chromatin organization, such as replication, and where here doubling matters. So here approaches and lessons. Then I will move to the issue of histone recycling that we will have already touched on with replication, but now in a transcriptional context. And then hopefully we can sum up about what we learn and what we wish to learn and how this can help us to better understand their importance and dynamics in the context of cell fate. So just as a mean of introduction, and you've heard about this before, if we consider the chromatin landscape, now it's important to see this in 4 dimensions with both the 3D organization and the time. And clearly it's a versatile system that underlie cell fate, identity, and disease. And as depicted here, you have a different level of organization ranging from the basic unit, the nucleosome, up to the organization with the positioning in the nucleus. And just to remind you, with the basic unit, of course, the histones that are a key component of the core particle. And at that level, we already know that there's a lot of things that can happen and that can be changed. And that can potentially also impinge on the higher order organization. I just want to give one example with the centromeric variant, CENP, which is key for the organization of the centromere of importance for chromosome segregation. So having said that, we now also learn, and you heard about it before from Amos Tanai, that the organization in the nucleus has to take into account the chromosome territories, the TADs, and the chromatin fiber as much as we understand. And so basically the challenge we are facing now in each cell is to understand how you maintain or change a particular state during DNA replication, repair, and transcription. And here, as I mentioned, there's all these aspects, the DNA modification, then the histone composition involving histone chaperone, the histone modifiers, the histone readers, the remodeling factors, and non-coding RNA. So here we will focus on the histone variant and chaperone. And so clearly there are multiple scales to consider in space and time, and there's now a lot of possibilities to access to information using both omics and imaging approaches. So here in my talk, I would like to illustrate a little bit more the imaging aspect. And so here you can try to understand how this organization can change during the cell cycle, during development, and in different environments. And I would like to stress one point that is the difference between what I call signaling and epigenetics, because you can use the same type of modules. In one case, signaling can be short-term, and in the other case, epigenetics is more stable, propagated through multiple cell division, and in some cases, through generation in an organism. So here, if we consider the major bricks with the histone H3 variant in mammals, so what we're going to talk about, you can consider them also as Lego. So here you can see they fall into these families with the replicative variant, H3.1 and H3.2 here, that differ from H3.3 by 4 or 5 amino acid. And the most extreme variant is Senpei. So they were actually discovered by their different migration properties on Triton acidurate gel by Alfred Zweidler in 1977. And a key aspect in terms of their properties is that they are expressed in a manner that is controlled during the cell cycle. So the replicative variant have an expression that peaks in S phase. In contrast, the other variant, so the replacement, H3.3, is expressed throughout all phases of the cell cycle, G0, G1, G2, and S phase. It has been connected with transcription and also very important for major chromatin rearrangements, such as those observed at the time of fertilization, when the sperm is getting rearranged, removing protamine to bring in histones. Then the most extreme variant, as I mentioned, Senpei, is expressed in G2. So the only one which peaks in S phase is the replicative one. So now if we look at the replicative variant versus the replacement variant H3.3, one striking feature between the two of them is that the core particle look very, very similar. So one may wonder why do you need, or what is the purpose of these different variants. So this was a crystal structure obtained by Hirota Shiwana in Hitoshi Kuromizaka's lab. So, as I alluded, the idea of brick and architect, how you build a nucleosomal landscape. So here you can see it's a daffodil made in Lego, which has been made at the occasion of the Curie fundraising. So it's present at the Pantheon right now. So it's huge. And this is for us quite an interesting opportunity. But I think that in the same way as this big daffodil has been built, you can also see how you can build the nucleosomal landscape over the chromosome distribution with a particular genomic location. So with the the replicative variant, which can be found at any place, genome-wide. Then you have, and it's deposited by the CAF1 complex during replication. Then you have the replacement variant, H3.3, that is found enriched at regulatory elements, active genes and promoter, but also at gaps where potentially in a transient manner, nucleosomes have been lost. And this is deposited via a complex called Hera. And then you have also deposition of H3.3 in other places, repetitive region, such as region in the telomeres and others involving the DAX-ATRX complex. And finally, for the centromeric variant deposited at the centromere, normally it does involve the chaperone H-jerk. So you can see that there's this tight interplay between the H3 variant and the distinct chaperone to build up this organization that is quite sophisticated. So if we get back to the variant and their differences, you can see that they are here in the histone 4 domain, major differences. So this amino acid in the replicative variant with this motif that we call SVM that differ from the AIG found in the H3.3 variant. And in the amino terminal tail, you have at the position 31 this difference with an alanine that is replaced by a serine in H3.3. So what is quite interesting is that this motif in the histone fold is a recognition motif for the chaperone that are involved in their deposition. And so the SVM motif is key for CAF1 to deposit the replicative variant. And concerning the AIG motif, it's key for the recognition by UBN1, a subunit of the Hurry complex, and DAX is part of the ATRX DAX complex. So basically, a key difference between the variant is their mode of deposition. But is that all? So that was sort of the question we had in mind, especially considering the function in various species when considered at the level of a whole organism. If we consider Cerevisiae, there's only one non-centromeric H3 variant, which is mostly like H3.3 as an ancestral form. Yet introduction of H3.1 works better to humanize budding yeast because there are some additional adaptations. So this is quite interesting a model. Then in Tetrahymena, the lack of H3.3 impairs viability. Doesn't impair viability, but leads to sterility. So here it's not absolutely required for development. In C. elegans, the loss of H3.3 is viable, but it leads to stress sensitivity. So here again, not an absolute requirement for H3.3 development, but a particular role in the context of stress and also during replication. And there's also this link with sterility in Drosophila, where, again, not an absolute requirement for development. In Arabidopsis thaliana, here, in contrast, it's lethal, so it's absolutely required. In mouse, the complete loss is lethal, but there's also, for partial impairment of one or the other of the copy of H3.3 effect that affect the development. And in human, attraction to the H3 variant was really brought about with the observation of the pediatric glioblastoma, where mutation in the H3.3 variant were found associated with aggressive cancer. So clearly that was raising the issue of why and how important they could be during development. But as I alluded to, the problem was that in many instances, there's the issue of protamine exchange. And so to disentangle a role of H3.3 from the one related to the replacement of protamine is not simple. And so here, the Xenopus system came up for us as a very useful tool, because in Xenopus, you do not replace the H3 variant by protamine in sperm. And so you can bypass this issue at early stages of fertilization to explore potentially other role of the variant. And so this is what we did. And we used an approach with morpholino. And so the morpholino are basically antisense RNA that can interfere with the endogenous RNA targeting specific protein of interest. So here we did it to target specifically H3.3. And as you can see in the movie, with the wild type injection in the control here, as opposed to what you can see when you have the downregulation, you have here a proper gastrulation. Whereas here, there's a lot of death. You cannot close the blastopore, and it really is terrible. However, you can rescue this phenotype by co-injecting wild-type mRNA encoding H3.3. And this only works when we have this rescue with H3.3 itself. It doesn't work if we inject H3.1. So we've been using this system then to play with the rescue to try and understand what was key for this rescue of this gastrulation defect. And it turns out that what was key was not the deposition pathway per se, but one amino acid in the N-terminal tail. And so this is what David and Katya considered in their way, looking at this embryo, that regardless of the deposition pathway, the amino acid 31 in the variant H3.3 was essential at gastrulation in Xenopus. So, well, I just want to say, and you heard about gastrulation earlier on today, the quote from Lewis Wolpert, it's not birth, marriage, or death, but gastrulation, which is truly the most important time in your life. And so think about it for yourself. And so now the take-home message is certainly that H3.1 and H3.3 are not simply redundant. And here we found a key role for one amino acid in H3.3 N-terminal tail, S31, which can be phosphorylated. But I'd like to stress that of note in plant, conversely, alanine at position 31 on the replicative H3.1 is key for genome stability. So this is just a paper that came out in Science, and I would like to invite you to check out. So for both of them, this amino acid is quite important. So even if structurally the core particle look the same, this variant can contribute to genome function in different ways. And I would like also to stress that using various systems can be really interesting, depending on the biological question that you want to address, to get insight into the function of factors that can have evolutionary counterparts. So now if we get back to the histone-based nucleosome versatility and this core particle, as I mentioned, we have now to take into account the histone variant incorporation, so the replicative, the centromere, and H3.3, which is particularly important in active chromatin. But as I said, there's an importance in the amino terminal tail of particular residue, and this amino terminal tail do harbor lots of histone modification. And so there can be potential crosstalk with this modification, and this is something quite interesting to look into, considering the different post-translational modification that we know of, with K4 trimethyl, K27 acetyl, largely associated with active chromatin, or K9 trimethyl or K27 trimethyl associated with repressive chromatin of different type. So I've pointed out this particular region, but as you can see, this K27 region is not far from this residue. And in the context of the H3.1 in plant, the modification by monomethylation on K27 is actually quite important with a crosstalk with the alanine on H3.1. So now moving to the other part of my talk that has to do with replication. So this is a time that is certainly challenging the maintenance of chromatin states. If you consider during replication at a nucleosomal level, what happens with the passage of the replication fork, you have disruption of the nucleosome head of the fork with transfer on the newly replicated DNA. So in this case, you have, for one disruption, two new nucleosomes behind that have to be done. So there's a need to have also newly synthesized histones to be brought in a de novo assembly pathway to get a full complement after the fork. And so this does ensure to restore nucleosomal density. But it's important also to consider how this is challenging histone-based information, both with the pre-existing modification, the potential variant, and how mixing old and new material can function in terms of dosage. So here the question is how you coordinate recycling and de novo deposition with replication for progression. This has been a question that a number of labs have contributed, as I've listed here, and we are still learning quite a lot. So in terms of the de novo nucleosome assembly pathway, we had learned a lot with approaches using in vitro system initially. And this is a simple summary where concerning the variant during DNA replication, the replicative variant is deposited in a CAF1 mediated pathway in a manner that is coupled to the replication fork. Given that CAF1 can interact with pCNA, that enables deposition with the progression of synthesis. And then for the H3.3 variant, it's Hurry or DAX that can ensure deposition in a DNA synthesis independent manner. And so that occurs through potential connection of Hurry with POL2 at genes and regulatory elements, but also at places where there's a transient nucleosome-free region with an interaction with DNA potentially. And then for the telomere and pericentric heterochromatin, it's ensured via DAX ATRX mainly. So this was for de novo deposition in the context of the progress in replication. The work started mainly with Anya Groth and Armel Corpet in my lab, and Anya has made really tremendous work to characterize in more detail what happens with the recycling of histones, the connection with the MCM machineries in replication, and the role of, we showed also the role of ASF1 in this context. So you can check out for more the reviews from Anya. But now one aspect is how can you track new and old histones in vivo? And so here I would like to illustrate for you a tool that we've been using quite a lot and that has a lot of potential. It's the SnapTag system. So basically, the SnapTag is an enzyme moiety that has been modified to be able to make covalent link with benzyl guanine derivatives. And in this way, if you have a SnapTag that is linked to your protein of interest, here we've been tagging H3 variant, you can then, by using your benzyl guanine derivative that is labeled with a fluorescent moiety, here TMR, you can visualize your histones. And so you can see here a visualization. And what is nice is that you can do a pulse and it's also permeable dye and you see the labeling of your cells. But you can also play with the system by pulsing first the histones and then you chase, which will then enable to synthesize new histones that will not be labeled. And in that case, what you see is the old histones that were present at the beginning of the experiment. But you can also play with the system by first blocking using benzyl guanine derivative that is not fluorescent, in which case you quench what is present to begin with. Then you can chase, and then the newly synthesized histones will come in. And then if you pulse with your labeled TMR, then you will be able to detect the new histones. And that is what you can see here. So this is really a very convenient system. So here by imaging, but we've now also developed approaches to follow genome-wide by a chip the presence of the SnapTag. So basically, with this tool, what we wanted to do is to try to question this issue of memory and trying to see if there's recycling of H3.3 during the process of transcription. And also to try and learn which chaperone is in charge of the old histones. So at least what I mean here is the histones that were present before transcription. So to do that, what Julia in the lab did, she used an approach where she could label transcription with EU labeling. And she did that in a system where she had either the total H3 or H3.3 labeled, then she would chase, and then she could look at old H3.1 or H3.3 by having a pulse with TMR at the beginning. And so she can do that with or without transcription using clavopyridol as an inhibitor. And as you can see here, you have here the EDU incorporation. Following the inhibition of transcription, you see that it is basically largely dropped. And you can look at the labeling of H3.3 here. And when we quantify this, we see that old H3.3 is really rapidly lost over time in cells. And when we inhibit transcription, we see that that is not the case. So transcription does cause a loss of H3.3. So then we wanted to look at where H3.3 can be located in chromatin. So we use this approach where we can quantify the signal corresponding to the factor of interest and compare it by segmentation to a second signal. And so in this way, we can have, when we simulate data, a shape that is indicative of colocalization in blue, or anti-colocalization when we do the quantification with this various area by segmenting the primary signal and compare to the second. So using this approach with a staining for Hera or H3.1 or H3.3, and then the phosphorserine 7 of polymerase, what we could see is that H3.3 nicely showed colocalization with POL2, while H3.1 did show the pattern of anti-colocalization. And we also saw that there was quite a nice pattern of colocalization for Hera with POL2. So using this approach, we then tried to look at the dynamic of new and old histones with the system I mentioned before. So now for the new histones, we do the quench first, then the chase, and then we label with TMR in a pulse. So when we do that and do the comparison of POL2 and new H3.3, what we see is that when we've done the chase for 2 hours, we see an increase of H3.3 relative to the site of POL2. So now if we do the same thing for old histones, now what we see is just the opposite. So we see a loss of old histones. Sorry, so this is indicative that there's a dynamic exchange. So you get new histones and you lose some of the old ones. So now the question is, which chaperones does take care of this old H3.3 on chromatin? So now, using the same type of assay, we can interfere with a chaperone that could potentially be involved with siRNA. And so here we did that for the Hera chaperone. And what we could see is that when we deplete Hera, we diminish the loss of H3.3 in the context of transcription. So here, clearly, Hera ensure the maintenance of a significant proportion of old H3.3. So then the question is, which partner of Hera participate in handling new and old H3.3? Because is it simply the reverse of the new deposition? As I mentioned, for the new deposition, it's this complex that does it. So we know of the different components in quite some detail, thanks to the work of many people, including in particular the work of our colleagues in Japan, Toshio Senda, who found the trimerization as well of the Hera complex. And so what we did is we did SI for the different partners. And so when we do that, and using the assay I described before, we confirm that if we do an SI against UBN1, this is affecting the new histone deposition. And also if we do an SI against ASF1. So this is all fine for the new deposition, consistent with what we knew before in general. But now, looking at old H3.3, to our surprise, the SI against UBN1 had no effect on H3.3 loss, and the SI against ASF1 was the sole one giving rise to a loss of old H3.3 as compared to the SI for array. So then we went on, and given all the knowledge we had from the structure and for the importance of particular amino acid for the interaction with the different partner, we introduced for rescue different mutants. So mutant of array that would be defective in the binding to ASF1, or mutant in the binding for UBN1, or mutant in the capacity for array to trimerize. And here we looked again at the new deposition, and the rescue for new deposition works beautifully with the factors that can bind to UBN1 and that also trimerize, but not when we have the mutation. And conversely, when we look at old H3.3 recycling, here the interaction of array with ASF1 is absolutely essential, as you can see on this figure. So basically, what we can conclude at this point in time is that the process of transcription is responsible for eviction of old H3.3, and part of it is recycled. And this is entailing, of course, a balance where there's new H3.3 and old H3.3, important for the restoration of nucleosome density. And so this is how we depict as new H3.3 deposition. This pathway where the complex of array involved its trimerization and UBN1, and ASF1 can be bypassed in this context. And then there's the recycling that we now can put in the picture that function in a different manner, where here it's array and ASF1 that are really critical for this reincorporation. So of course, this is really tantalizing to think about how this works and what are the consequences for the way we want to think about it. And what you can see is certainly that array here can act as a hub to coordinate new H3.3 deposition and old H3.3 recycling. But this is raising a series of questions, because if there's a co-transcriptional H3.3 recycling, why? Old and new H3.3 have in fact fundamentally different PTM patterns. And I just want to remind you here that you can find the high level of methylation on K4 on old H3.3 in the transcribed region. In contrast, on new H3.3, there's a very limited number of methylation modification that could be detected, and only mono and dimethylation at K9 have been reported. So this shows that potentially there's a way to distinguish also modified version of new H3.3 and old H3.3. And so the idea we would like to think about is that the chromatin recycling in cis at pre-existing or proximal site could direct the maintenance or reinforce the active state of transcription. However, ectopic recycling could introduce change potentially. And so this could be possible if we think of the site serving as a nucleating site for other modification to be propagated. So that could operate in trans or perhaps distant sites. And finally, there could be also retention in the subcompartment in cis or in trans, so in which case there would be more of a loose maintenance via histones available in local subnuclear domain. And so that is probably also something important to bear in mind, where the localization within the nucleus of this domain may find their importance. And I would like to finalize now, just to summarize, why these distinct variants, replicative and replacement. So there can be shared role different sources and pathway to maintain genome integrities, but also specific roles. So with the work in Xenopus showing the specific H3.3 function at the time of gastrulation with phosphorylation of the S31 locus on H3.3, but with the H3.1 in plants, as I said, for genome integrity maintenance. And for new and old H3.3, why different pathway? Maintenance versus switch of chromatin states, way to fine-tune chromatin. So these are questions we wish to get at. And also the fact that you mark the genome with these different variants can enable to distinguish particular region. And this is something I will tell you more about in the next talk, next week, whereby H3.3 and H3.1 boundaries themselves, so the region where you change from one to the other, can be important for early replication initiation. And so I'd like to stop just to stress that incorporating different variants, new and recycled histone is part of a wider landscape. You've seen all other aspects of the chromatin organization in the nucleus, and they are all in a constant crosstalk and interplay with each other. So yes, this is a sort of the image of the elephant that you touch from different places. So based on the old parable from Jalal Rumi, who was discussing how blind men could see different things when they touched an elephant. So let's continue touching our elephants. And I'd like to thank the people that have been involved in the work I described. Camilla has a strong interest in the ongoing questions. Audrey, Jean-Pierre, Dominique, and Ivan, along with David and Julia and Guillermo, have been involved in the work either on the Xenopus system or on the imaging of the transcriptional aspect. And with Alberto, we've been working quite a lot on the genomic approach that I will discuss later. And of course, the role of our friend Tosha Senda and Ronan Marmostin for the structural aspect of the Hurry complex. Antoine Coulon for his help in the modeling of the imaging data and our platform for imaging and sequencing. And of course, our funding agencies. We could not do our work. And I think you can see everybody making this little sign, which is friendship. And Stefano, who would love to be with you, but has been hit by the COVID, is here as well. And so thank you for your time and interest. Happy to take your questions.
Thank you very much, Geneviève, for beautiful talk. So the talk is open for questions. Any questions from the audience? Please go ahead. Can you speak loudly, sir? Can you see us, Geneviève?
I'm trying, yes. So as you know, it's true, in mammals, there's H3.3 A and B genes. They have the same protein composition, but they have a different expression control. And so it would be, and it's something that we are interested in, is to try, it's been actually something we have entertained discussing with Eran, to tag respectively one and the other with a different type of tag. And so that could be done with snap and clip, for example, to try and see whether the one deriving from one gene or the other have distinct potential distribution. But we haven't done it yet, but that would be really, really cool.
Any more questions? I would like to ask you a question, knowing your interest in cancer, and after you deciphered so nicely the system here in normal cell, what happens in cancer cells to these different variants? Are they keeping the same rules as normal cells?
So, I would say it could be different in different types of cancer. And you have here in this very audience people that are very interested in glioblastoma, where there are mutation in the H3 variant in particular. And so there's all this area of the oncohistone landscape. So initially it started with a K27 mutation, but there are more mutations that have been found, and even some that can interfere with the remodeling of the nucleosome core particle, as shown by Sigal Kadosh. So there's clearly aspects related to changes in the balance and the dynamics of the H3 variant. From what we have done, one thing that was particularly striking to us is the importance of the change with a very increased level of SNP, so the centromeric variant in many cancer type. This increase in SNP level is something that we have been looking into in more detail. And in this case, there are changes in SNP localization. So it doesn't go anymore only at the centromere, but goes also in the chromosome arms. And so this is potentially changing a number of things with respect to the cells, and it does affect cell fate. Well, in a model system that we have been using, we could see that the overexpression of SNP could promote the transition into EMT. That is something that we found quite exciting, and we are trying to understand a bit more about it.
But I was meaning mostly to the chaperone. Do you see different chaperones and co-activators or co-proteins are coming that are putting the different variants differently in the cancers? Yes. Not about the variants themselves, but the chaperones.
Yes, actually, this is correct. There is actually work showing that there can be changes in the way the variants are sort of handled by different chaperone in the context of particular cancer. So in the context of the transition to metastasis, there's a change in terms of also the switch from the CAF1 to the Hra pathway. And even in what I described before, the SNP deposition ectopically does depend on the DAX complex, not the HGER complex. So there's really an interplay where the balance and the choice of the chaperone can change when you are in the situation of cancer. That is absolutely true, but it could be different, and this is something we need to understand better for different types of cancer cells.
Eran.
Beautiful talk, Geneviève. So histones themselves are relatively stable and long-lived, and yet only a partial of the histones are recycled. So how is it regulated, and what happens to the histones that are not? Are they recycled later, or what's going on there?
Well, let me repeat. So your question is why there's only one part that is recycled and not all of it? That's
and how is it regulated?
Okay. So the fact is that this is a question that we are not we don't know yet how this is actually regulated in great details. What I think could be important to bear in mind is that the histones themselves can be damaged. They can be oxidized, and so you may actually want at some point to get rid of them. And so for the moment, we don't know whether it's only the ones that are perhaps bad or not good anymore that are discarded, or if it is also part of the process to ensure that you keep a certain amount for a memory of the previously existing state. So this is really what we would really like to understand.
No more questions. Let me thank you very much again, Geneviève. And we listen to your second talk on the end of the week. Thank you very much.
Thank you.
So it's my pleasure to introduce our director, Eran Meshorer, who is going to tell us about CAPP-I links pluripotency with RNA metabolism.
Okay, you hear me? Yes. Thank you, Judith. What I didn't notice when I planned this meeting about, a year and a half ago is that I just came back from teaching 2 hours, and for some reason I put myself just 1 hour after this this lesson, so I have no voice basically. But we'll go through this together. Right. So in the context of Episystem today, I thought that it would be a good opportunity to talk about this work that is spearheaded by one of the Episystem students, Giuliani, and I also mentioned some of our other student, Patrick. But my lab is really interested in stem cell epigenetics and neurons, and we're doing mouse work where we're basically studying chromatin and epigenetic regulation during early differentiation and reprogramming, as well as human stem cells, iPS cells, and embryonic stem cells, where in the past several years we've taken an approach to basically model human diseases, mostly polyglutamine-related diseases, and the first publications are now starting to come out. But today I won't talk about this at all, and instead of focus on one of the proteins we identified. Another subject that we started analyzing in recent years together with the lab of Liran Carmel is a topic we call Paleo Epigenetics, which is epigenetics of ancient genomes. And I think in the second part of my talk, I'll talk about this topic, but again, mostly I'll be talking about one protein that we identified here. Of course, I don't have to already mention it, that embryonic stem cells are derived from the developing embryo at the blastocyst stage. And the approach that we took is a kind of a 3-direction approach to do biochemistry, cell biology, and in silico approaches to identify really the factors and mechanisms that operate during early embryonic stem cell differentiation. And today I'll just, I have one lecture, so I'll tell you about our cell biological approaches. And the idea was to generate an endogenously tagged fluorescent library in mouse embryonic stem cells. And it was actually an idea that was originated in the lab of Uri Alon, and together with the Alon lab, we kind of adopted it to work in mouse embryonic stem cells. And what we did was we took 3 reading frames of basically a viral backbone, incorporating a fluorescent protein sort of non-directedly into the genome. So it's not completely random because retroviruses, they have some tendency for genes. But what we do is we incorporate YFP. So this YFP has no start, has no stop, and no promoter. So if it's integrated somewhere inside the genome, nothing happens. This is 99% of the cases. But every now and then it will be incorporated inside a gene, usually inside intron 1 because it's the longest intron. And then we get a fusion protein. And then we can sort, if this protein is expressed, we get a fluorescent cell, and we can now sort single cells into 96 square plates and expand the surviving clones. And this work was done by Ed Puganti, Raghu Ram in my lab, together with Hari, both of whom are actually now in Florida. And we recently published this work. So we generated clones of YFP, and we did the same library in cherry. So in each one of the clones, you have a different proteins that is endogenously fluorescently labeled. And although we don't consider these proteins to be functional, we actually consider them not to be functional unless proven otherwise. But at least in over 90% of the cases, when we compare their localizations, their endogenous localizations, to localizations of the antibodies that we found or in the literature or whatever, so you can see these pairs. These are our YFP clone, and this is the antibody either from some catalog or from our own staining. And really in like 25 out of 26 cases or so, we found perfect localization. So these proteins, even though they are not functional, they are very useful for screening because they report the correct expression and localization of the endogenous protein. So we started doing some screens with these libraries, and one of the simplest screens that we did is basically look at loss of fluorescence during the early differentiation. So we start with an expressed protein. It's green or red, and then we differentiate the cells with retinoic acid, and we look what happens. And we did this for both nuclear and cytoplasmic. So this is just an example from the nuclear screen that we did. This is a protein called SET that we published a few years ago. And you can see its loss here with imaging. And the nice thing that because all these proteins are fused to YFP or to cherry, you can use a single antibody to basically identify your protein by Western blot or by IP or by chip or but whatever. So you have a bait for any of the proteins in the library. And you can see here the loss of SET. And this was actually quite surprising. It was a novel, if you will. It was a known protein set, but it was not known in the context of embryonic stem cells. And the loss really mimics the kinetics that you see for pluripotency factors like OCT4, nano, and others. And this was really unexpected. And it turns out that actually SET has two isoforms. And we're only labeling one of them. So the YFP was inserted here in intron 1 of SET alpha, and we're completely blind to SET beta. And this was the reason that this was previously overlooked, because if you look at both of them using other proteomics or RNA sequencing or whatever, microarrays in the old days, you're basically looking at the sum of the two transcripts, and you don't really see SET alpha. And only SET alpha is really lost. So SET beta is expressed. In fact, it is even increasing during differentiation. So I think this is why it was missed. So this library also allows some unexpected discoveries. We also developed this, this is I alluded to in my first slide, this in silico approaches that we've taken was basically a website that we generated called BindDB. You're welcome to use it. Where we collected all the ChIP-seq data sets in embryonic stem cells, both mouse and human. The idea was to perform a reverse ChIP in silico. If you want to know what binds a particular promoter, you can try and pull down your promoter with no success, or you can just do in silico. In embryonic stem cells, there are so many maps now that you can just punch in your promoter or 100 promoters, and then you can retrieve whatever binds there or is enriched relative to background. And actually, we were able to really identify the different proteins that are bound to SET alpha versus SET beta. And we found that SET alpha is really bound by pluripotency factors, whereas SET beta is just poised. So you can see it's bound by RNA pol 2, but not by the others. So it's really specific, despite the close proximity of these two promoters. So again, I'm not going to talk about SET. This was just a teaser for the nuclear screen. It was recently published by Patrick Lim, who's actually is a little bit under the weather today, so we told him not to enter the hall, but he's here virtually somewhere. So SET, we found that SET regulates early embryonic stem cell differentiation. It's actually essential for naive pluripotency, and by interaction, proteomics, we found that it interacts with beta-catenin. But I want to talk about the cytoplasmic screen, which was kind of more outside our comfort zone, but yet a postdoc in the lab decided to go and chase cytoplasmic proteins. And he identified this interesting protein called caprin-1. And what we found was kind of a twofold difference. One is a slight decrease, which was not too impressive, but also kind of a relocalization. You can see these kind of foci that appeared somewhere during differentiation. Now, caprin-1 is it was a known RNA binding protein, and it has these RGG domains, RNA binding domains, and it interacts with RNA. And one of the things we found was that, I hope it will work, yes, so that when we looked at our time-lapse imaging, it really formed these really beautiful condensates or aggregates immediately after mitosis. This looked like RNA granules or stress granules. And just to see whether this is the case, we poisoned our cells, either with arsenate or with the proteasome inhibitors. And in both cases, they formed these stress granules. And indeed, caprin-1 was immediately recruited to these stress granules. So caprin-1 is expressed in embryonic stem cells, slightly downregulated, and is recruited very nicely to these stress granules if you either stress the cells or immediately after mitosis. We also did some localization with known stress markers like DDX3, X, G3BP2, TIA1, and we found co-localization. In fact, this also exposed us to the wonderful world of stress granules. If you notice here, for example, you can see that only one of them is caprin-1 positive. It seems that the whole world of stress granules that is not entirely understood, but which is really specific to some proteins. So you can see that most of them are really co-localized, but every now and then you see stress granules that are specific to one of the two proteins, and it's not understood why. Okay, so the next thing to do was to knock out caprin-1 in mouse embryonic stem cells. We used CRISPR. We got a very nice knockout. It was relatively easy, and the ES cells don't care about loss of caprin-1, not to the extent of morphology or growth. They did show some growth retardation, but really they were pretty happy in the undifferentiated state. So these are two separate clones. However, when we tried to differentiate the cells, we saw complete defective differentiation in whichever differentiation lineage we were trying to take the cells into. So defective cardiomyocyte differentiation, ectoderm differentiation, and endoderm, and even if you just do either embryoid bodies or retinoic acid, you get this skewed differentiation. And this can be rescued by a cDNA of caprin-1. We also did RNA-Seq to see what's going on. In undifferentiated cells, very few changes. In early differentiating retinoic acid-induced cells, very large changes. Again, suggesting that retinoic acid is really required somewhere during early differentiation of embryonic stem cells. And when we looked at what these RNA-Seq results are telling us, we found that in retinoic acid cells, of course, in undifferentiated cells, there's very little
data to process, but in early differentiating cells, we found this RNA processing, RNA metabolism, RNA processing. So this already was quite interesting. And then we decided to look for the coprin-1 interacting proteins and RNAs, so coprin-1 interactome. So for this, we basically, grew undifferentiated embryonic stem cells and early differentiating cells. We lysed the cells, and then we split them. We IP'd with coprin-1 antibodies, and then we split them into protein and RNA. And here we mass spec these proteins using LC-MS, together with our collaborator Newman Shea, who was then in Singapore and now moved to Canada. And we basically did also RNA sequencing, which will give us the RIP-seq portion of the interacting RNAs. So when we did go gene ontology functional analysis of the RIP-seq data, okay, these are the RNAs that are bound to coprin-1. We found that in embryonic stem cells, we get this like a general RNA localization and splicing and processing, but then in retinoic acid, we suddenly switch to differentiation and development. So whereas there is this generic role of coprin-1 in ES cells, as the cells differentiate, it seems to be interacting with RNAs that are suddenly specific to differentiation. And when we did this experiment, this is a validation experiment doing coprin IP PCR, we found that some of these interacting RNAs, such as SOX9, were actually upregulated in the coprin-1 knockout embryonic stem cells. And this kind of raised an idea that maybe coprin-1 is actually involved in basically in RNA processing or basically degradation of these RNA transcripts. So we started doing just a case by case. We took one of those cases like SOX9, which we found to be upregulated in the coprin-1 knockout cells, and we did alpha-amanitin. And in the one or two cases or three cases that we checked, it seems to be the case. This was just a control that didn't show any differential expression. And then we decided to go genome-wide. Sorry, before that, we wanted to see whether SOX9 RNA is really localized with coprin-1 protein. So we've done single molecule RNA FISH, and actually it was very hard to see the colocalization. So we repeated this under stress conditions, and indeed this was the case. And this was not the case for again, FOXP1. So we went genome-wide, and we did the same experiment. So what we're doing here, we're treating the cell with alpha-amanitin, which blocks RNA transcription. And then we take time courses. So every 2 hours or so, we collect the RNA. And then together with Michal Rabani, who is an expert in RNA degradation kinetics, we did modeling of the RNA degradation assays, and we looked for global changes in RNA degradation. And what you can see here is a shift. So when you compare knockout to knockout, you see that it's centered around the core, but when you compare wild type and knockout 2 or knockout 4, you get this shift to the right, which means that you have more stability in the knockout, in the RNA knockout, or more degradation in the presence of coprin-1. To validate this, we teamed up with Miguel Esteban in Guangzhou in China, and they are experts in SLAM-seq experiments and other aspects of RNA degradation. So they did this experiment. We sent them our cells, the coprin-1 knockout and the wild type, and they basically, in the SLAM-seq experiment, what you do is you basically pulse your cells with 4SU, and then you chase them with unlabeled uridine for, again, 1 hour, 2 hours, etc. You collect the RNA, and then you can sequence the RNA, and you can basically look at all the new RNA by this Forest U signature. You can detect it with a chemical reaction. And again, when you compare the half-life of the copperin-1 knockout versus the wild type, you see that in the wild type you have a lower half-life, or meaning that in the presence of copperin-1, the more RNAs are degraded and they become more globally stabilized in the copperin-1 knockout. So the model that emerges from all this is that we have this mechanism operating in the cytoplasm, or in these granules, where copperin-1 is involved somehow in RNA degradation during early differentiation. We actually think about it a little bit like polycomb acting in the nucleus, degrading early differentiation-related transcripts, but only operating in the cytoplasm. However, copperin-1 is not a nuclease. So our next step was really to try and identify the potential ribonuclease which is involved in this process. So of course, we went to the mass spec again. This time we IP'd copperin-1, and we looked at all the interacting partners. We had a very nice and clean data. We got a lot of the known interacting partners, and really the functions that are known to be associated with copperin-1, RNA helicase. This is actually quite a novel direction that we're currently checking, but I don't have actually data to show you. This is a M6A pathway and a lot of RNA binding proteins. But in the others, we found one exoribonuclease called XRN2. So at first we were happy, but then we were sad because XRN2 is actually a nuclear ribonuclease, and copperin-1 I showed you is operating in the cytoplasm. So what's going on? So it turns out that XRN2 actually relocalizes to stress granules following stress. So you can see that copperin-1, XRN2 is indeed nuclear. This is nuclear. This is staining for XRN2 in ES cells, right? And this is copperin. So they don't co-localize really. But when you now stress the cells and you get these nice stress granules, and you can see now copperin-1 really now localizes into the stress granules, you suddenly see XRN2 also exits the nucleus and goes into these foci, and they now sit together in these granules. So this was really nice. And what was even more striking, that in the copperin-1 knockout cells, XRN2 is maintained in the nucleus. So copperin-1 is required for the exit of XRN2 from the nucleus into these stress granules. So it seems that copperin-1 XRN2 is somehow involved in this RNA degradation pathway that operates in early embryonic stem cell differentiation. And overall, just to wrap up this first part, is that I showed you this library that we generated in mouse embryonic stem cells. And the nice thing about it is we can really do screening in various directions. So I showed you one example of basically looking for loss of fluorescence, but you can now measure protein dynamics using photobleaching assays, and you have the proteins expressed at endogenous levels. You can look at heterogeneity, because these are all clonal, so they're derived from a single cell. You can do localization tracking, so changes in localization during early differentiation. You can identify proteins that are recruited to sites of DNA damage. If you just shine a UV laser, you can image your cells 5 minutes, half hour later. And of course, because of the fusion, you can basically IP any protein that you want with the proper antibody for YFP or cherry. Okay, good. So I have time left. And yeah, so our library is relatively limited because I have to say I kind of failed to be able to fund large-scale libraries. So we have a few hundred clones, but we are very biased to highly expressed proteins, because the way it works is that, of course, only expressed proteins will be fluorescent. So when we sort the cells, if we lower, you know, if we really lower the sensitivity of the FACS and we get all these very lowly expressing proteins, we start getting garbage also. So we were really just looking at the proteome that is highly expressed in the S cells. So we're so far satisfied because there is you know, more opportunities than what we can bargain for. But if you want to really do it large scale, you know, I tried to recruit funding, but I couldn't at some point, so I kind of quit. But then I decided that maybe it's too technical. So we have about, I don't know, 250 maybe clones. Yeah.
You mentioned that Caprin is also sort of the stress granules, or the colocalization is also formed right after mitosis. Is it related to a function with the stress granules, or in that case it's not?
Yeah, we are wondering about this. I don't know the answer. We are actually looking into it, but one of the ideas that we're entertaining is that there is a phase during the cell cycle where the cells are particularly stressed, and that is immediately after mitosis. Whether this is true or not, I don't know, but we can really see this relocalization of Caprin-1 into stress granule.
So I have a question also. Where is the specificity? So it goes to all RNAs?
No. So there is a specificity. Thank you. When you look, there is a sequence that Caprin-1 binds. It's actually very similar to the polyadenylation and cleavage signal, AAAUA, or AUAAA, I don't exactly remember. But it's not completely identical. So there is some kind of specificity, but in a way that it can really degrade a lot of transcripts. So I don't have a full answer to this. I mean, we analyzed this, and this is what came up from the sequence data. It seems a relatively redundant sequence, but not redundant enough to be present in all transcripts. But again, when you look at the number of transcripts, these that are associated with Caprin-1, we have thousands. So it's not degrading one or two. It's potentially degrading 2,000, 3,000 transcripts. Yeah.
So Eran,
That's it. Yeah.
maybe it's okay for people to ask you
Yeah,
questions
sure,
after
sure. Yeah,
this
yeah,
talk, and
of
then
course.
maybe
Yeah, that's
we'll
what
go to the
absolutely.
other one.
Right.
Any?
Yes.
Can I ask now?
Yes.
Sure.
Please.
So the fluorescence library you created is specifically in RNA, right? So you mentioned one example that was alternate site that's cause of unlabeling, and maybe there would be in translation of like truncated protein you have labeling but not function. So how do you overcome this problem?
So I guess you're asking is if we have an insertion and we're generating a truncated protein, our YFP doesn't have a stop. So only if it's inserted in frame, you will get a reading frame. So we are only getting fusion proteins, no truncation protein. Sometimes, of course, there are, I guess sometimes you can fall in between and generate NMDs or something like that, but we usually just don't see the protein, right? So if we get a fluorescent protein, it's usually because it's inside the reading frame. This is the only way we can get a protein because we don't have a start and we don't have the stop. So we are, right? We're
Maybe it will affect some of the non-coding sites and
again, it could, but if there is some, usually if you have a truncation or anything to do with the processing of the RNA, you usually don't get a protein. There is NMD or something that degrades the transcript already. So we don't see it. We're not going to pick it up because we're only picking up fluorescent cells. And this means that we generated the fluorescent protein. I'm not saying that we don't have sometimes, insertion next to a promoter with some weird reading frames. We do have a few of those, but we throw them out. Because
Eran?
we basically sequence everything. So, yeah.
Eran, I was wondering whether when looking at the protein that you found in your interactome, so in the others, I noticed that there was PARP1 and also NPM1. So do you know how this protein, which are also nuclear, how they interact with caprin?
Yes, we have several nuclear proteins. Yeah, we were wondering. So our first guess was that caprin maybe is shuttling between the cytoplasm and the nucleus, and we were actually planning to test it, but we never got to it. So I don't know if caprin is actually shuttling or not, and if these guys never exit the nucleus, then I don't know the answer. But of course, they are present at least for some part of their lives in the cytoplasm because they are generated there. So every protein it has to be made in the cytoplasm before it enters the nucleus. So, but I don't have a good answer.
Yeah, what I was actually thinking is whether when you have DNA damage, for example, if that is also triggering some particular type of association that would appear, because I thought what was really nice in what you showed is that the association with XRN2 was detected upon stress. But what about the others? Perhaps it's something similar.
Yeah, I don't know. we haven't tried DNA damage yet. Actually, the other protein that I showed, SET, is involved in DNA damage, but we're too young in the caprin-1 world, so no answer.
Opening a lot of possibilities.
Yeah, thanks.
I noticed that in the retinoic acid treated plants, the caprin binding RNAs were enriched for mesoderm related terms. Do you think there's a bias against mesoderm?
Yes, so Atilgan was asking about differentiation bias, and we do see differentiation bias. So I think that there might be, I don't know, the problem is that we've done these experiments, because the experiments are so wide in terms of the genome-wide data that you get, we've done them only with retinoic acid. So we didn't really do lineage differentiation, and to answer this properly, this is really what we need to do. Because retinoic acid might have to do some with the bias that we see, but we do see a differentiation bias, but I don't know if it's related to the protocol. Oh, okay. Please.
So in the mass spec data, you mentioned and you showed that there are several YTH family, domain family proteins that probably interact with caprin, and those also have roles in RNA degradation. So are there any kind of phenocopies between, or at least transcript half-life? That's a better established role for this.
Yes, so we were trying to phenocopy caprin-1 knockout with XRN2 knockout. XRN2 is essential for any cell, and it's impossible to knock out. So we're doing knockdown experiments, and actually the knockdown phenocopy okay. The problem is the levels of knockdown. So if you knock down too much, they're completely unhappy. But we've tested so far XRN2.
And with the YTH, those?
I don't know yet. Yes, I don't know. Please.
How do you know that your fluorescent addition does not alter the protein activity?
We don't. I consider all proteins not to be functional. we use these only for screens. Once we identify protein, we go back to the endogenous protein. Yeah, it's only for screening purposes, not for functional purposes. And if you want, sometimes, if it's really important for us to test if the YFP fusion, then we can rescue a knockout with it. If there's a, if there is a phenotype for a knockout, you can rescue. We did it once with the HP1. I know it's functional. Yeah. Okay.
So, second part.
Okay, thanks. All right. I have less time than what I planned, but it's fine. Less questions also. So the second part, I'll tell you about this topic that emerged in recent years, which we call now Paleo Epigenetics. And I'll tell you about the recent work. And this was done really in a very close collaboration with the lab of Liran Carmel and spearheaded by David Gochman, who's now just established his lab at the Weizmann Institute. He was then a PhD, MSc in my lab, and then PhD in Eran's lab, and we did it together. So in recent years, as you know, there's a revolution in ancient DNA, works coming out from the labs of David Reich and Svante Pabo, mostly basically were able to sequence ancient DNA at very high resolution. And basically we had the DNA of the present day human and several apes or non-human apes, but in recent years, about 10 years ago, these sequences came out, the Denisovan from the pinky finger of a young girl, and now we have about 5 genomes of Neanderthals, but at very high resolution, very high coverage. And when these sequences came out, it was clear that they are almost identical to humans, and that sequence alone cannot explain the major differences that we see anatomical, etc. So we were thinking about, and there are, these numbers are not even correct. You have to divide them by 2. So sorry for the, because these numbers really change with every sequence that comes out. So we have less than 50 fixed amino acid substitutions between the modern humans and the archaic ones, and about 20,000 fixed non-coding. So of course the non-coding genome is where it's all happening. And the idea was, can we say anything about gene regulation in archaic genomes? And then we turn to DNA methylation. Because, of course, you know that DNA is methylated by methyltransferases. And we will hear Howard Cedar on Tuesday talk about it in depth, one of the pioneers. And DNA methylation is directly controlling gene expression. So when enhancers and promoters, especially in CPG promoters, are methylated, they suppress gene expression, and this comes in the context of CPG methylation. And when you want to determine DNA methylation, you basically bisulfite your DNA, meaning you deaminate your cytosine into a uridine. And the concentrations that you use in the lab will prevent a methylated cytosine to be deaminated. And then you can, of course, compare the sequence before and after bisulfite, and you can compare used to C's, and you know if your C was methylated or not. But if you take your DNA and you put it 100,000 years in a cold cave in Siberia, the same process will take place, right? So it's just not very practical for a PhD, but you could do it, right? If you had 100,000 years. So actually 2 natural processes take place. One is degradation, so the DNA is fragmented, and the other is deamination. So this is the natural degradation process of DNA, and your C deaminates into a U, but also your methylated C deaminates into a T. So there is this asymmetry that happens. Now, during the preparation of the libraries, at first, both labs of Svante Pabo and David Reich, now David Reich is now not doing this anymore, but they both did a process of uracil trimming. And this actually what kept the signal in the sequencing. So this allowed us to do what we did. Because of the uracil trimming, what you get is the following. Here is a pre-mortem DNA sequence of, let's say, a Neanderthal. This is a methylated region, this is an unmethylated region. Now the person dies, and deamination takes place. So some of these methylated Cs will turn into Ts, some of these unmethylated Cs will turn into Us. And then of course now you pick it up from the cave, you extract DNA, and then during the library preparation, you remove the uracils. So what happens is that in a methylated region, you have a very high C to T conversion ratio. And in the low, you have a very low C to T conversion ratio. So you can basically compute this C to T ratio. And what you get is a high C to T here and a low C to T here. So we basically just took the sequence of the Neanderthal, and you ran a sliding window of something like 50 CPGs, and you need a lot of statistics. So these sequences, you need at least like 30x coverage in order to get enough statistics. But it looked like this. Right? So this is the C to T conversion ratio across the genome, and it gives us a proxy on DNA methylation. At the time, there was no methylation of humans in bone. There were 26 other samples, but not bone. So we measured it using a high-throughput bisulfite sequencing, genome-wide. And we found really a remarkable agreement between our measured DNA methylation. So you see that methylation is almost binary. This is unmethylated, this is methylated, etc. And it looked very similar. Right, so one of them is the Denisovan, one of them is the Neanderthal. I don't remember which is which, but it doesn't matter. This is modern humans, right? And really they seem to be identical. So this now allowed us to focus on the differentially methylated regions. Okay. We actually expanded our collection to include several chimpanzees as outgroups. So again, we didn't have bone methylation, we had to do it. Present-day humans, anatomically modern humans. This is very important because these anatomically modern humans died 5,000, 10,000, 20,000, sometimes 40,000, 50,000 years ago, and we can reconstruct their DNA methylation to make sure that there is no bias in the method. So it's exactly the same method, only on modern humans. So what we were looking for are to identify what we call evolutionary differentially methylated regions, where in modern humans, whether reconstructed or measured, it will be either high or low, and the opposite in the archaic ones. And of course, the same with the chimpanzee that will provide us as the outgroup. So these are modern human specific, what we call evolutionary DMR. So these are the different options. These are the modern human specific. They can be archaic specific, Neanderthal derived, etc. So we focused on modern human derived because it's also the most interesting, but also from a statistical point of view, they're the most robust. We identified 873 differentially methylated regions. So this is compared to about 40 genes that are altered in terms of sequence. So much more. And we were able to assign about 550 differentially methylated genes by distance, and actually by measuring the three-dimensional or reconstructing it, we were able to assign 70 additional ones. So this is just an example how the data looks like. This is a measure of differential methylation, and suddenly you see a peak in a specific gene. And when we took all these genes and we wanted to ask what is their function, we found a very strong enrichment here. in the voice box. In fact, less than 2% of the genes affect the voice box, but we found that 11 out of the 20 most differentially methylated genes affect the voice box. This is a very, very strong enrichment. And I'll show you one example. It's a gene called NFIX, and this is the differential methylation map. So you can see green is non-methylated, red is methylated. You can see here the Denisova and the Neanderthal, the chimp. And here are modern and ancient humans combined, so you can see that they are similar. So this is one example of a DMR, which is actually nicely reverse correlated with expression if you look in several different tissues. So the idea that we thought of is that we have high NFIX activity in our archaic friends. And somewhere here we have medium expression of NFIX. And actually there are loss of functions mutations with NFIX in Marshall-Smith and Sotos syndromes. These are NFIX loss of function mutations. And what you get is this retracted face and changes in the voice box, in fact. So we decided to go a step further and to generate Neanderthal NFIX overexpressing mice and to ask what happens to their voice box. So we generated the NFIX overexpressing mice, and we had this recording device. This work was done by Malka, my lab, together with the lab of Nikki London. So mice speak in syllabi. Yeah, this is they sound like birds. This is ultrasonic vocalization. So they speak in these syllabi, which look like this, and people who are interested in mouse songs and speech, they try to interpret these, but I leave that to other people. So we collected something like 12,000 vocalizations over a period of, 2.5 years. Malka, correct me if I'm wrong, maybe 3 years. And what we found, so this is an overlay and projections of all the ultrasonic distribution. There are more than 3,000 in each group. And what we found that there is a change in the NFIX overexpressing mice. There is one change here and another actually frequency that we never found in the wild type mice. So this is quite interesting. So we think that NFIX seems to be controlling at least one of the genes that are controlling facial features via its expression. And what you see here is the NFIX loss of function. This is the silhouette of a modern human, and this is the silhouette of a Neanderthal, and this is the level of NFIX activity. Okay, so this work was done in collaboration with many people, as I said, with Liran Carmel's lab and spearheaded by David Gochman, and currently by Malka in my lab, and with collaborations all over. So I'm not going to list everyone. So it's here. And the first part was really began by Gajendra and now taken over by Giuliani. And thank you. There's going to be time
So questions to the second part, please.
There was a point a bit earlier in the presentation where you were speaking about differentiation of expression between the different groups. How can we know expression of specific genes in the ancient humans that we...
Right.
Yeah.
Okay, so this is what we see here is not the expression itself. It's just what we think is that methylation, at least in specific regulatory sequences, controls expression. Okay. Now, because we cannot measure the expression in Neanderthals and archaic genomes, we use DNA methylation as a proxy. But sometimes DNA methylation is not related to expression. So, in order to ask whether this particular DMR or this particular enhancer or this particular promoter is really regulating expression, we look across different tissues. So what you see here is the reverse correlation between the mean methylation and expression of NFIX in different tissues of modern humans. Okay, so we believe that we can, yeah.
Yeah.
Okay.
Do you have any idea how did they sound like? I mean, they have lower voices.
No.
Does it mean that they didn't talk?
No. So actually, it's a riddle because there is the hyoid bone is so soft that it's one of the only missing pieces from the Neanderthals. We don't have it. So to reconstruct the vocal cord, the vocal voice of the Neanderthal is very, very difficult from the current anatomical pieces that remain. And therefore, these questions are not answered. But the overall anatomy is quite similar to humans, right? So I mean, they don't have a chin. We are the only apes with chins, and this reflects the anatomy of the voice box. But otherwise, the idea was to see whether there is a change. I think they probably would have had the capacity to speak. I don't know if they could, but they, from an anatomical voice of view, probably the answer is yes.
Probably a very silly question, but it's not related to where the pieces of bone were taken from, right? Like other bones, a femur, a tibia, finger, we all have the same DMRs?
Yeah, so you can see it here. In modern human bones, there are a very large collection. So this is femur, this is teeth, this is skull, and we made sure that they all cluster together. Here we have toe, femur, finger, and several from the apes, and we made sure that they all cluster together. So it's just a division between these two groups. So we believe that it's sex independent, age independent, and limb, tissue independent as well. Yeah.
So maybe it's not relevant, but I think because the Neanderthals is very interesting, they vanish and then Homo sapien takes the come over. So, and you mentioned about the differences of the voice. Is it relevant to the language? And then because we have a language, maybe they don't form a specific language.
Yeah, it's not that they vanished and we lived together with Neanderthals, but we survived, right? There were tens of thousands of years that we lived in parallel and we made children together. We are part of us is Neanderthal. Between 0 and 3 or 4% of each of us has some Neanderthal lineage. Again, these are all speculations, so I don't know if one of the ideas, and you can read it in Yuval Noah Harari's books and others, that it was the communication between modern humans that made it possible to survive all the other groups. But we don't know. We don't really know. In terms of brain size, their brain was bigger. So if anything, we were less intelligent.
So thank you very much, Eran.
Sure.
We should. So we reconvene at 4:00. So I guess we should start. our last session for the day and our last talk. And I would like to introduce Omri Wurtzel from Tel Aviv University, and he is going to talk on M6A is required for resolving progenitor identity during planarian stem cell differentiation. Omri, please.
Okay, so thank you and good afternoon, everyone. It's all working. Okay, I'll start with a disclaimer similar to Eran that I also came from teaching, so I hope that you can hear me all the way back. So what I have in plan is first give you an introduction, a pretty lengthy introduction to the field, then tell you about what I think is the most important question in the field of regeneration using model organisms that was resolved over the past decade. And lastly, we'll see how much time we have for that. I'll move to speak about a project from our lab, and I hope that I'll have enough time, but if not, then we'll leave it a bit open, and it's fine because it's a school and learning is an ongoing process. But let's start with regeneration. Regeneration is the regrowth of missing tissues and organs, a process that is relevant to essentially any living organisms, at least any organism that would like to obtain functions that it has lost. This loss of function could be the result of injury, of chemical damage, aging. It could be a consequence of diseases, and it's relevant to essentially every organism. It's a process that has captivated both aficionados, scientists, and naturalists for centuries, even thousands of years. And here I brought two examples from the 17th and the 18th century of organisms that were documented in nature as being highly regenerative. And that, of course, wasn't during a time where a systematic study was done in a lab setting, but the study of the process of regeneration is certainly one of the oldest, as it remains a very curious and unresolved scientific field. If we look at different animals, animals that we use, for example, in the lab, we can see that they vary very dramatically in their ability to regrow missing organs. So, for example, the adult nematode C. elegans is entirely postmitotic, except for its germline, and therefore it doesn't regenerate. It cannot recover missing functions following injuries. Other organisms, for example, different types of salamanders, can grow certain types of organs. For example, limbs, tails, parts of certain internal organs, etc. Some organisms can regrow organs in a much more broad manner. For example, planarians shown here that can regrow essentially any missing tissue following an injury. These processes, so they vary between organisms, both in the scale of the tissues that can actually regrow following an injury, and also in the dynamics of the response. If the growth of a limb can take months, in some organisms, growth of missing tissues can happen in a matter of days in a highly predictable manner. Okay, so there's a lot of desire to use the right model organisms to get at these questions. Now, obviously, one of the most important questions is understanding whether this entire process has some kind of a common evolutionary history. So I have here a very simplified phylogenetic tree that includes the bilaterians. Those are the animals that have bilateral symmetry, right and left, but not dorsal and ventral, and some non-bilaterians here. And so if we want to be more specific, those are the deuterostomes, protostomes, including insects, nematodes go here, lophotrichozoans, including the mollusks, annelids, and platyhelminths go here. And here are some more basal bilaterians, such as acials. and those are cnidarians. If we'll take this very simple phylogenetic tree and we'll overlay regenerative organisms, what we'll find is that we can easily identify multiple different species that are found in each of these phyla. Okay? So those organisms, they vary very significantly in terms of their tissue complexity, in terms of their lifestyle, in terms of their lifespan. So we cannot really point to one particular trait that correlates well with the ability to regenerate missing organs. I can say for sure that not all of these animals have the same capacity to regrow missing organs. So let's take a couple of examples. This is a spiny mouse. It's not your usual lab mice, and they can regrow certain connective tissues that other mice and other mammals are completely unable to recover. Instead, they'll develop a scar. Axolotls, they are well-known vertebrates, model organisms for studying regeneration. They can regrow their limbs, certain parts of their CNS, and other parts of their body, but they are not able to regenerate their entire body. Here in cnidarians, they can regenerate all of their any missing body parts. So those are examples for animals that can do whole body regeneration. And specifically in my lab, we use two different model organisms that can do whole body regeneration. One of them, which is the more traditional model organism for studying regeneration in the lab, is planarian. It's a platyhelminth, and it can regenerate any missing body part. And here, this is a type of chordate, Polycarpa meteligera, which is an ascidian, and those can also regrow any missing body part. So those animals, you'll remove their brain, they'll grow an entire brain, or in this case, a ganglion. So what's the advantage of working with two distinct organisms? That's a question that I'm also trying to understand myself. It is interesting and fun, and also importantly, other than that, being interesting and fun, is that those are very, very different in terms of evolutionary history. I mean, there's more than 500 million years of evolution between those, and one, of course, of the advantages of using model organisms that are really different in terms of their evolutionary history is that we can try and understand what are the differences and the similarities in regenerative strategies that those are using. For example, is there something that is common to all regenerative organisms and might be missing in mammals? Or is it different strategies, two different paths that leads to really spectacular ability to regrow missing organs? Okay. From now on, I'm going to focus on planarians. Polycarpa is a more, we're working with planarians for the last 2.5 years, right? Something like that. 2.5 years, and essentially everything that we do in these animals is new because it hasn't been used in the lab except between us and the lab of Professor Noashenkal from our university. Okay, so I'm going back to this slide, and I would like to point out that even in the 17th century, they were able to identify planarians as being extremely good at regeneration. So those are planarians. I'll go back. Okay, those are planarians that live in fresh water. These are land planarians, but this is a trait that we can find throughout this family of animals, that they can regenerate very well. And indeed, the ability of these animals to regenerate has captivated the interest and curiosity of many scientists. And probably one of the best-known scientists to make modern research something that is a possibility in planarians is Thomas Hunt Morgan, one of the most prominent heroes of modern genetics. So for many years, he worked with planarians and with Drosophila melanogaster, and for his work with Drosophila melanogaster, he received the Nobel Prize and made numerous contributions for the study of genetic analysis. Here, this is part of a manuscript where he shows how cutting planarians to small pieces results documenting their growth of their bodies over time. Now, what they don't tell you when they show you this, usually they don't tell you, is that he stopped working with planarians at the end of the 19th century because he said that their genetics doesn't make any sense. Which is, I mean, I can see that, been working with planarians for a while, but I mean, part of it is that, part of the reason, it's not a joke, part of the reason is that many planarian species are not diploid. And doing genetics, especially at the turn of the 19th century, is extremely difficult with organisms that are not stably diploid. Of course, in the lab, we use species that are diploid and we can work with them. And the advantage of actually using a simple model organism that is amenable to different lab techniques is that we can study this process in multiple resolutions. If we think about regeneration not as just some kind of a therapeutic goal or endpoint, it is a process that is similarly complicated to embryonic development, and as such it has to be studied at multiple resolutions, from the molecule all the way to the complexity of the organism. And using a simple invertebrate model that can actually regenerate very well allows us to do studies at those multiple resolutions. And I think that the next slide shows how well and how predictable the regenerative capacity of planarians is. So you see here a knife that goes down and cuts a planarian into pieces, and with each time that it cuts it, it generates multiple fragments. And by the end of this experiment, there are 5 different fragments, and those 5 fragments, very predictably, within 14 days, I mean this is within 14 days, but within 7 days, they'll complete the regenerative process, and that means that they will regrow their heads, will regrow their tails, regrow all of their internal tissues. And that means that during the course of roughly 7 days and not months, which is something that other regenerative organisms require, over the course of 7 days, we can go through almost all of the stages of tissue growth and analyze them. Okay. Other advantages for using this model is that they're easy to grow. You can grow them in simple plastic boxes in the hundreds and even the thousands. So a lab can have an operation where it has tens of thousands of animals for experiments. They're amenable for gene function study by feeding them with double-stranded RNA. So that's very simple, and then we can feed them. In a single experiment, we can have dozens of experimental groups, having each group fed on different kind of double-stranded RNA to test the functions or analyze the functions of multiple genes. And finally, there is a large research community. Planarians are by far the most, the largest scientific community working on regeneration using invertebrates. So that's also something that's very, very important in order to be able not to inventing the wheel for every method that we use. So what does this process of regeneration look like? This is a photo of planarian, and you'll see a red line where I amputated its head and then had a time lapse of the head growing back. Okay. So this is the cut, and within 7 days, the entire head sprouts in a process that is generally referred to as epimorphosis, the growth of the tissue from a blastema. By day 7, there are already eyes, and if we'll use some kind of a technique like whole mount in situ hybridization, shown here, we can identify the regenerated organ. So, for example, we can identify here, labeled in green, I think it's synapsin, labeling the newly formed brain. You can see here the intestine. You can see eyes, and of course we can see many other organ systems. So it captures a lot of the complexity that we have in the process of regeneration. However, it's fairly simple animals to work with.
Does it have memories or some kind of learning?
That's a good question. I mean, it does have some sort of reflexes. For example, aversion from light. There are many controversial papers dating back to the 50s, 60s, and 70s from several groups that report on different memory essays or learning essays and paradigms, and those haven't really been repeated, and they are heavily criticized. But and I bet that they do have some kind of memory.
So can you tell if it happens to the new head?
To the growing head, and if I'd have to bet, the answer would be no. But since there is no really established paradigm for doing these kind of essays in planarians, there is no good answer for this question. Yeah.
Will they regenerate if you do the cut?
Oh yeah, they'll regenerate. So I'll answer. So what kind of cut can you do so that they wouldn't regenerate? You can't do. They'll regenerate from anything. The only place that they can't regenerate is if you'll take a part of their pharynx. That part is not going to regenerate. I'll show you why. Okay, last question before I continue.
Just the part about the pharynx and it doesn't regenerate. Is it with like the common sound and vertebrates, like Siemens type?
Oh, the reason is that the pharynx is unable to regenerate other parts is that the pharynx doesn't have stem cells. Okay, that's the reason.
Basically, to dissociate them to single cell.
Nope, that's not going to work. So if you'll dissociate them into single cells, and then you'll try to reconstitute some ball of cells, it's not going to regenerate. However, Hydra and Idarian, if you'll do that to a Hydra, it will be able to reform its original body pattern and effectively regenerate.
What's the minimum mass, minimum cell amount that could be regenerated?
It's a very small amount. It's roughly 10,000 cells, but it's not about the number of cells. It's about the thickness of the tissue. In order to regenerate, the animals have to be able to form morphogen. This is the model. They have to form morphogen gradients of Wnt and Wnt inhibitors from one side of the axis, one end of the axis, and the other end of the axis. If they can't do that, they will still regrow tissues, but the tissue that they will regrow are not going to be the tissues that they need. Okay? So they are not going to form this predictable head and a tail. They'll form, for example, a head and a head. Okay? Okay, so let's move forward. Wait, I have this here. So what is actually required for regeneration? This is something that it's with a really small disclaimer. It's relevant to essentially any regenerative process happening in nature or any regenerative organism except, for example, Hydra. You need the ability to grow new tissue, no matter how, whether it's stem cells, the differentiated cells, whatever. But you need some ability to grow back tissue. You need the ability to have morphogenesis, to form the new organs, and you need to integrate the new organ with the old organ. Okay, so for example, you're regrowing a brain, you need to have it rewired into the existing body. So those are really three essential aspects of regeneration that we see in almost every regenerative organism. In planarians, the cellular source for growing the new tissue are stem cells, adult stem cells that are called neoblasts. So you see those neoblasts here labeled in blue, and you can appreciate that they are extremely abundant. Roughly 20 to 25% of the cell number in planarians are planarians adult stem cells, and those are generally called neoblasts. Neoblasts it's a name that has a history going back to the 19th century, and we use it to this day to reflect on anything that can divide in the planarian body. Once you irradiate the animal with ionizing irradiation, it causes DNA breaks, and the dividing cells, they die. Okay, so this is an untreated animal, has plenty of stem cells, plenty of neoblasts. This is the irradiated animal, doesn't have any neoblasts. Okay, now when those two animals are being challenged to regenerate, this is simply by amputating them. Okay, so cutting, the untreated animals will always be able to regrow missing tissues, in contrast to the irradiated animals that will always fail to do so. So the stem cells are essential for producing the missing tissues.
But wait a minute, it continues to live, the irradiated one?
Oh, so the irradiated animal will live for roughly 7 to 20 days, depending on the conditions. It will die once the epidermis starts to break off, because it is being continuously maintained by this population of stem cells. So these stem cells, they're not only responsible for regeneration, they are also responsible for tissue replacement. So they will live for a while. Okay.
What's the stain?
This is labeling for a stem cell marker that we use that is essentially PIWI, which is, I think that in the mammalian world it's used for as a germline marker, but in planarians, in nidarians, this is it's considered a universal.
So there is no
The blue PIWI.
So there are no stem cells in the head.
In the head tip there are no stem cells, and in the pharynx.
The pharynx we can see, but the head tip.
Yeah, the head tip as well. They have, there is a region that doesn't have stem cell, and those regions are not going to regenerate. Okay.
You said pharynx.
Yeah. So the pharynx is not going to regenerate, and also right at the head tip, it will dissect this section. Other parts of the body, if they have stem cells, they are going to regenerate.
Can you inject stem cells from one animal to the other?
This is in a couple of slides. We'll have to wait for it. Okay. Okay. But of course, the stem cells themselves are not sufficient for having productive regeneration. So this is an instance of productive regeneration. An animal that was cut a while back, regenerated a head, regenerated a tail. However, it's very simple to produce animals that can regrow tissues, but their regeneration process is not really productive. So for example here, this is simply by inhibiting beta-catenin, the effector of the Wnt pathway, we can generate animals that have multiple heads. So you can see a head at the original anterior and a head in the posterior, and also what appears to be, I hope that you can appreciate that there are eye spots here, appear to be also heads in the lateral end of the anterior part. Okay, so regeneration is not only about forming new cell mass, it's about forming the right cell mass, having it patterned in the correct manner, and having it integrated to the tissue in the correct manner. Okay, so that actually leads me to, if I'll try to put it in very simply, is that the key to regenerating well is to produce the required cells at the required anatomy at the right time. Okay, that's it. Not too much to ask. So if we have stem cells in the posterior, we'd like those to produce tissues that are usually found in the posterior, for example, intestine. However, once the animal is being injured, so here I'm showing an amputation, this part of the tail will now have to generate a new anterior fragment. It will have to regenerate a new head. So stem cells from this region will now have to form a massive amount of neurons and similar and cells that exist in anterior structures. Okay, so there has to be some kind of method to determine the identity of the cells that are being generated. Okay, and one of the for many years, one of the biggest debates in the planarian community was what is actually the developmental potential that planarian stem cells or neoblasts they have. Can they form any kind of cell type? Are they restricted in their identity? There could be cells in the posterior that can form either neurons or can form intestinal structures, but they would be just differentially active depending on the regenerative status of the animal. So the two models that existed, I'd say probably until 2011, when a couple of very important papers came out, was that either the tissues are being formed from lineage restricted neoblasts, or another option is that pluripotent neoblasts generate all of them, all of the different cell types. And of course, there could be combination of those. Those are not mutually exclusive models, but those were by and large the models that that were heavily considered. Okay, so questions: are neoblasts pluripotent individually or collectively? Do we have one cell that can form everything, or since we know that they can regenerate any missing body part, they are just collectively pluripotent? Yeah.
Could it be cross differentiation event?
So in planarian, there isn't differentiation event. It is being shown by papers that consider this possibility by using a radiation kind of experiments.
Okay, but you said that the statement you showed before are adult stem cells.
Yep.
Isn't it contradictory to the right model system?
To this one?
Yeah.
I mean, adult stem cells. I didn't say that these adult stem cells, and at that when this image was taken, it was unknown whether these adult stem cells have a limited developmental potential, like you'd usually expect from stem cell systems, or whether they are pluripotent. That was an open question. Okay, so I mean, there are maybe multiple ways to approach this question, but I mean the paper that gave a conclusive answer to the question whether neoblasts can be individually pluripotent came out at 2011 from the Redin lab, and the experimental scheme is simple enough. Technically, this is really heroic work where animals were taken and stem cells were isolated out of them. Okay, probably reduce this process. Something that its optimization took a couple of years to just this animation. But animals were taken, and then stem cells, or what they thought are stem cells, were isolated by using flow cytometry. And those were used as donors. Then other animals were taken, and they were lethally irradiated. So following lethal irradiation, there are no stem cells in the animal body, and they cannot regenerate at all. The obvious next thing to do is to inject these single stem cells or transplant them onto the irradiated host. This is extremely difficult technically to handle those cells in a way that maintain their viability, and in terms of getting them into the tissue and having them grafted there. But once you have this protocol and the cells actually graft there, then you can wait, allow the stem cells to recover and to grow a colony of cells within the organism. So this is what you see here. And then examine the colony. If the entire colony has just a single fate, like what's shown here, then most likely the stem cells are lineage restricted in their developmental potential. But the other model, would suggest that maybe this colony will have multiple cell type in it, and in that case, the colony, depending on the number of lineages that you can analyze, this colony could be pluripotent. Okay, so this is the experiment, in a nutshell. So this experiment was done. So we'd like to assess. They like to assess neoblasts in a colony, can find out whether the colony is lineage restricted or pluripotent. And this is from a 2011 paper that shows that, this is a colony. In green, this is the stem cell marker, and in this blow-ups, you can see co-expression here in magenta and here in yellow of two markers. So this marker here is a chat, So it's a neuronal marker, and here in yellow, this is GATA456, which is an intestinal marker. So what they shown in this in situ is that there are colonies that have cells from both ectoderm and endoderm. Okay? So based on this analysis, the colony is not restricted to just a single lineage, at least 2 lineages. Okay. Question?
So we talked about so many difficulties in performing this experiment in vivo, but is there a reason why this experiment can't be done in vitro, trying to differentiate the cells for different
Okay, so first of all, we don't have a method to grow planarian stem cells in a culture, and I'd also argue that, with several caveats that this experiment has, is that if you'd like to study the developmental potency of cells in vivo, it's of course better to do that in an in vivo setting in the natural environment. Put it into some kind of medium, and who knows what happens to the cells when they divide. Okay. And I'm saying it, and this experiment as well has some caveats. I mean, you could argue that taking a cell out of one organism, transplanting it, it's also something that induces a lot of stress on the cell, and it could have adverse effects. Okay, but it did have really nice controls to
It's
that.
like HSP, like
Yep.
hematopoietic
Mhm.
method.
Yep. Okay, so we have here the analysis of 2 lineages, but what's even more, the most beautiful result that came out of this paper is that these transplantations, some of them in a low frequency, didn't only enable the formation of multipotent colonies. Some cells grafted in the hosts and completely rescued them. Okay, so a single stem cell was transplanted into a host that has hundreds of thousands and actually millions of cells, successfully repopulated this entire tissue and allowed it to regenerate. So based on this result, this was a 5% rescue using this approach. So based on this result, there was very conclusive evidence that under the limitation of this experiment, planarian neoblasts could be individually pluripotent. Okay, so this is a conclusion from here. Okay, but then actually, it doesn't mean, of course, that all of the stem cells are pluripotent, right? That's clear. I mean, that at least 5%, because there was a 5% rescue. Okay, but then at about the same time, multiple labs in the planarian community, let's see. Okay, multiple labs in the planarian community came out with, there was really a string of papers. I mean, those are, I'd say, probably 20 to 30 papers, all in all, that reported that neoblasts have a specialized gene expression. And what it means is that they could identify stem cells within the animals that express transcription factors that were associated in a one-to-one relationship with a certain lineage. So let's take this one for example. They identified, this was I think 2015, a population of neoblasts that expresses MyoD, transcription factors required for formation of muscles, and inhibition of MyoD resulted in the progressive deterioration of the muscle tissue. Okay, and of course it led to the deterioration of the MyoD expressing neoblasts. Okay, so there is a certain population, there is actually a very large population of planarian neoblasts that already expresses transcription factors that are associated with differentiation to distinct lineages. And I mean, that's quite, that was quite bizarre, considering the fact that some neoblasts are pluripotent. We don't know how many. And another, actually, it's not a caveat in the experiments, but we actually don't have any defining characteristic of the pluripotent stem cell other than its ability to form all of the lineages in a transplantation experiment. Okay, so we know that this exists. We know how to functionally analyze them, but we don't have any characteristic for a pluripotent stem cell. That was, in the community, was a big question. So what is the characteristic of those pluripotent stem cells? And of course we can modify our model and say that perhaps there are lineage restricted neoblasts, but they are being fed by a pluripotent neoblast. I mean that's fine. Okay, that's another good model.
I wanted to ask if when you do the transplant, you just take the random cell and hope it can live out?
I mean, well, when they published the paper, they thought that they had a method to enrich for neoblasts. I mean, and also the logic behind it makes sense, and the people who did it are very careful in doing these kind of experiments. However, I mean, it's been a decade since, and now we know that if you'll isolate cells randomly, you'll get to a very similar success rate in transplantations. So, I mean, they thought that they had the method, but I mean, I think that the largest factor to the success of their experiments was the fact that they identified the anatomical region into which you can transplant a cell without it being lost inside the intestine, which is widespread in the animal. Okay? Okay, I'll continue a bit and then I'll take some questions. Okay, so that's one model, and of course this model actually raises many new and important questions. So for example, which of these is self-renewing? I mean, we know that the pluripotent stem cell is self-renewing, but are these self-renewing? Are those something that is similar to a transient amplifying stem cell? And because we always like to make those analogies between different stem cell systems that we know. And so those are all open questions, and I'd like to propose in two models which were, I mean, that was open. That question was, to me, that was the most important question in neoblast biology, and it was sort of resolved last year, I think, in a very satisfying manner. So one model is that pluripotent neoblast, the pluripotent neoblast produces lineage restricted neoblast, and those, they could either be proliferative or not, but they form this other, the differentiated cell types based on their identity. And the other model is that pluripotent neoblasts just transiently express some factors that we associate with the lineage, but it's not commitment. It's just part of its process in forming asymmetric progeny. I mean, you're pluripotent. I mean, the way that I'm imagining that you're pluripotent. You go into cell replication. You start expressing. I mean, we can identify markers of the epidermal lineage, but it's just because that this time the pluripotent stem cell is going to form an epidermis cell. Okay, that's it. So no commitment. That's the other model. Okay, and a reminder: only about 5% of this neoblast transplantation resulted in the rescue of the host animal. Okay, I mean, I think that this number, I mean, you could use it to justify why either one of the models is actually the right one. So we'll need some, a different manner to test whether, which model is the correct model. How much time do I have? 10 minutes? Okay, I have like 45 minutes. I'll take the 10 minutes. Okay, no problem. Okay, so let's see about let's focus on this transition from being pluripotent and being specialized. Okay, I'm not going to call it lineage restricted. It's specialized, expressing a factor that is associated with a certain lineage. Okay, and then what I actually like to show you is a very early analysis. This analysis dates back, I think, to like 2016 or 2017 that we've done, and actually shed a lot of light on this transition. I mean, and that was actually the first time that I found single cell RNA sequencing actually useful. Sorry. Okay, so each dot here represents a cell, and the color of this dot represents expression of any of the known specialization markers. And what you can see here immediately, that there is a bunch of cells that are not expressing a specialization marker, and a bunch of cells that many of them, a larger proportion is expressing specialization marker. And I mean, I should have hidden that, okay? Because when we've done the data analysis, we try to figure out, why some of the cells are appear to be non-specialized while others appear to be specialized. And I mean, at some point we did a cell cycle analysis, which is pretty trivial. I will also validate it by sequencing cells from labeled with hosts that allowed us to isolate cells that have either 2 copies of each chromosomes or 4 copies, so that translate to G1 or G2 cells. And when we've done that, what we saw is that most of the cells in G2 express specialization markers, whereas most of the cells at G1 don't express specialization markers. Okay. So this is what the single cell analysis showed. And that was really nicely recapitulated by in situ hybridizations. So cells from G0, G1 gate, in fact, were isolated, cells from S gate, cells from G2M, and each time that those cells were isolated, they were labeled with a different specialization marker, and they were always labeled with a neoblast marker. And this heat map is actually not for sequencing data, it's just a representation of the many in situs that were done as part of this project. So what you see here are on the column represent cells from a particular gate, so G1, S, G2M, or G0, postmitotic cells. The color represents the fraction of cells that was identified to express the lineage marker that was tested by this in situ. And it's row normalized. Okay, so this is the fraction, and the colors are being row normalized. And I think it's very easy to see that cells in G1, both by single cell RNA sequencing, what I showed you, also by in situ, those cells are not expressing specialization markers, whereas when cells go into S phase and when they are in G2M, and definitely when they are already differentiated, we can find specialization markers. And this was also validated by transplantation assays and colony assays that are similar to what I showed you previously. So based on all of those experiments, what we concluded is that the stem cell, the pluripotent stem cell that is up here in the hierarchy, this is actually the stem cell that can maintain itself, can divide, and all of those cells just represent some transient states. And those transient states, I mean, those amount to, I mean, gets me back to this initial question that I was asking. The key to regeneration is to form the right cells at the right time. How does it happen? So part of the answer is that the stem cells can choose their identity which each time that they go through the cell cycle. Okay, so here a neoblast forms one type of cell, then when it goes back following mitosis to G1, it can form a different type of cell. So when the animal have this amazing plasticity in their body, because once the animal is amputated, the cells sense a different signaling environment, and now they have the opportunity to differentiate, to form a different kind of lineage. And I mean, that's not, of course, the end of the question that we have for studying biology of planarian stem cells of neoblasts. There are many open, big questions that we and others are asking. The evolution of neoblasts, whether it's a specialized cell type. I mean, some people think of stem cells as being something that is similar to a blastomere, but to actually be a specialized cell type, similar to germ cells that can form an entire embryo, but they are extremely specialized. So those could be there could be a very interesting evolutionary history to those cells, and they are found not only in platyhelminths, what determines proliferation versus differentiation ratio. And I mean, what I really wanted to end with is about the question of how cell identity is established, and from there move to a research project that we worked on in the lab, but I don't think that I have enough time for that. So with that, I mean, so we'll leave it to a different time, and Yarden, my student, has a poster on this project, so you can, I encourage you to go and talk to her. So thank you for your attention. We're happy to take questions.
Questions for Omri?
As you track this, Scott, so how many evolutionary conserved genes do you have between humans and planarians? And if so, have you checked if there's got any dates between the stem cells of the two species?
Yeah, so great question. So so planarians are as closely related to mammals as flies are. Okay? So in terms of signaling pathways, for example, you can find all of the signaling, all of your canonical signaling pathways in planarians, and you can find them in flies, you can find them in humans. In terms of pluripotency factors, so those are actually some of them are not conserved, but we're very, I think research community, for good reason, is very much mammalian oriented in terms of the way that they think of that. But if you look at an evolutionary tree, what appears to be the pluripotency factors that are conserved between different invertebrates, they're actually not conserved in mammals. It's mammals that have a different way of using pluripotent stem cells, not planarians are actually much more have much more conserved genes for that if you consider the entire phylogeny.
So the lineage restricted stem cells can go back to become stem cells, like we see
So
in
they are
the
not lineage.
intestine.
So they are
Yeah,
not
the
lineage restricted.
transient. No, the transiently expressing lineage specifically
Yep.
and go back to. It's like the trans-amplifying
Mhm.
cells in the stem
Yeah,
cell going
in
to.
the yeah. So I mean, you can think of that of a cell that divides asymmetrically. One of them will go on and become differentiated. Okay, so it will become, for example, intestine. The other one is a clean slate or blank slate. It can express different differentiation marker and generate a completely different cell type.
Excuse my ignorance on this, I don't really know very much about planarians. When they develop, do they also have the different stages of the embryo?
Okay, so when they develop, they do have different stages of the embryo. They develop inside egg capsules, and they have we have good staging of the different events of development. I mean, I comment on that there is a stage, and it's called S6. S6 where neoblasts first appear. So initially, when they develop, the cells don't have the ability of adult stem cells. They have the ability to form the embryo. Only at stage S6 in their embryonic development, they have cells that can be functionally compared to neoblasts.
So they have embryonic stem cells as well? Assuming. Have you ever
Yeah.
compared the two of them to see the differences?
I mean, those were sequenced, but there was no actual functional analysis that determined whether there are different factors that are required for very early stages of embryonic development, which I bet that they are. It's actually, I think it's also pretty technically complex because of the way that embryos develop in planarian egg capsules. But it's good idea.
Yes, please.
The model that you showed where you were showing like some of the stem cells based on like germ layers, like they have this vestibular bias. Is that mechanism, because you said it was done in planarian studies, so was it done only, I'm assuming this is all S. mediterranean, right? Or was it S. japonica as well?
No, this analysis is in Schmittea Mediterranean.
Yeah.
Yeah.
But between Mediterranean and and japonica, for example, as two regenerating planaria, do they have similar mechanisms? Because like in the case of like axolotl and salamander, like with the muscle regeneration,
Yeah.
there's two different mechanisms. So is it similarly conserved, or
Yeah.
are they divergent?
Yeah, so this is labs like Johan's Link lab that studied regeneration that studies regeneration in many planarian species. They have a collection of over 50 planarian species. So they determined that there are at least two key components to regeneration that every planarian must have, and that all planarian have, and that is their stem cells and their ability to use Wnt signaling in order to form the AP axis. Whether or not pluripotent stem cells are a component of each different species of planarians, it's not something that wasn't studied.
And just one short naive question. Because you mentioned cell cycle a lot. In planaria, what's the standard cell cycle? Because in axolotl and salamander, standard cell cycle is 48 hours, unlike 24 for most others. And in regeneration, like when you get the blastema in the wind zone, it actually decelerates and becomes 72 hours. In planaria, does it decelerate or accelerate, or is it still the same cell cycle?
No. So what happens is that I don't know to say whether the cell cycle becomes shorter or longer following an injury. What I can for sure say is that many more cells go into cell cycle following an injury. So there are very two predictable, prototypical waves of mitosis following an injury. One that is body-wide, the other one is located at the blastema. What could add information is that the cell cycle is usually short when you do a short EDU pulse. For example, you do it could be either a couple of hours. You see a lot of labeling at that point.
Great talk, thank you. Question: If you cut the planaria into two pieces and keep them close enough one to another, will there be a wound healing there, or?
Okay, so the question of whether it's going to be a wound healing is a question of what would you consider wound healing? When if you measure gene expression changes at the site of the injury, in case you allow, let's say that you make a big cut into the animal, but you don't remove any tissue. So that would be taking the animal and cutting it like that. Okay, so that's a big cut. If you allow the two flanks to come back together, you'll see within roughly an hour a complete shut-off of the entire wound response.
So it feels the other edge and just completes the tissue.
But in that case, I didn't say that there is going to be. It's not that the tissue was removed. It was just a cut. If you'll remove a tissue and will allow it again to attach, then you'll see that it's going to take time, but the animal is going to regenerate a wedge, for example, at the site of the injury.
Thanks.
Yeah.
So open wounding does have a role in maintaining the injury induced gene expression.
Very, very interesting. Thank you. So you talked about injury and amputation. Do the cells or the organs undergo aging processes or senescence?
They don't.
They stay young forever.
Okay, so with the species that I showed you, I showed you mostly cartoons, so it could be anything. But the species that I usually refer to is strictly asexual, so they will reproduce by fissioning. And so those animals never experience any sort of senescence. They replace. If you'll do some kind of an EDU pulse, you'll check the animals 2 months later. You'll see that all of your EDU positive cells are gone. Okay, so they will replace all the tissue all the time. However, the sexual strains, at least the one that I have experience with, at some point they cannot reproduce sexually anymore. Okay, but then there's a nifty trick. You can cut them; they'll regenerate new ovaries, new copulatory organs, and then they'll be able again it rejuvenates them, and they will be able to do sexual reproduction again.
Omri, since I'm the chair, I'll take the option to ask you, is it EPI meeting? Is there any EPI? The M6A is RNA, DNA, anything else that you know about histogeneration?
Okay, so planarians, like many invertebrates, they don't have DNA methylation. It was very unfortunate. When I started my postdoc, I thought that perhaps I'll find that they do have DNA methylation. They don't. And then, of course, the modification on histones play a major role. And these processes that we see in terms of specialization, you can also see that, for example, by using a TAC-seq or by using different kind of ChIP-seq with antibodies that you'd usually use in the context of mammalian development. So they do have that. RNA modification is, this is something that we are really invested in because it has major importances to the physiology of the animal. And once we do this functional analysis, we inhibit different components of the pathway. You'd see very, very dramatic phenotypes, both when you look at the entire organism and when you analyze the individual stem cell. You'd see that it cannot differentiate in the same way that it previously differentiated.
When should I cut it?
Cut it.
The director
Amputate.
says amputate. Thank you very
So
much.
Alright, thank you
Where is
to all
the
the
mom?
speakers, and we have a reception downstairs. Everyone is welcome.